Okay, I guess we'll go ahead and get started. Hi everyone, my name is Abby. I'm a graduate student in Wes Gruber's lab, and I have the pleasure of introducing Dr. Robert Carrillo for our seminar today. So Robert has a long history of studying how neural circuits are formed and how neurons ultimately find their correct partners, which allows an organism to evoke appropriate behavioral responses to environmental stimuli. So he completed his PhD at Yale, where he began his journey of studying synaptic development and plasticity in Drosophila. And there he described how electrical activity via voltage-gated ion channels and calcium signaling mediate synaptic refinement and specificity in motor neurons. He then went on to Kaizen's lab at Caltech as a postdoc, where he helped to describe a cell surface protein interactome in Drosophila that we now know instructs synaptic connectivity in several motor and sensory systems. He used the larval neuromuscular system specifically as a model to show that motor neurons in their muscles express distinct cell surface proteins called DIPs and DPRs that interact with each other to influence proper motor neuron to muscle connectivity. So today he'll talk about some of the work that his lab is now doing at the University of Chicago, where he's still interested in elucidating these molecular mechanisms that underlie synaptic connectivity, but also how these synapses undergo growth, pruning, and refinement changes once they reach their final targets. So with that, I'll hand it off to Robert to tell us about the fly neuromuscular circuit.
Thank you, Abby. That's a crazy summary of about 12 years of work, just boom. But thank you so much for the introduction. And thank you everybody for being here, even though I cannot see you. Okay, is that good? Yep, okay. So first of all, again, thank you for being here. It's great to virtually be there with y'all. I tried to postpone this as much as I could to see if potentially I could get a trip out to New York, but unfortunately, that's gonna have to wait till the next time. And you know, given these virtual seminars, I've tried to make it a tradition to figure out what the mascots are of the different universities that we give seminars at. And now I know that Columbia has the Rory the lion, which is very intimidating here. So now I picked up another one under my belt. So with that, let's get into the science. So today I'll be discussing examining synaptic connectivity and function in particularly the fly neuromuscular circuit. In putting this together yesterday, I have 120 some odd slides, so I may not get to the full function part of this, but at least I'll talk about the synaptic connectivity. And before I start talking about that work, I have to give credit where credit is due. As you all are aware, PIs, we don't do too much work at the bench anymore. I try to get in there when I can. But a lot of the work that I'll be discussing is from a graduate student named Yipu Wang, another graduate student named Micah Labrabi, as well as a senior scientist in the lab named James Ashley. And these are a couple other graduate students and research technicians. So to get a little overview of what I'll be discussing is I'll show you what the fly neuromuscular system looks like. And I'll hopefully convey that this is actually a great system to look at synaptic connectivity, even though it's not a neural circuit. It actually encompasses a lot of the properties of neuron-to-neuron interactions. I'll then discuss some of these proteins that Abby talked about, the DPRs and the DIPs, and how they may be underlying the synaptic connectivity. Again, if I have time, I'll be discussing this more recent project in the lab about synaptic plasticity in this convergent circuit where two motor neurons converge onto one muscle. And then finally, I'll give a summary and future directions. So to give a quick background, obviously it maybe wouldn't be a great synaptic or a neuronal talk if I don't give credit to some of the godfathers here. So this is, you guys all know Santiago Ramón y Cajal, one of the great neurobiologists in the late 1800s, early 1900s. And so he was one of the first to sort of postulate that the nervous system isn't a reticulum, that there are discrete units within the nervous system. And he used this Golgi stain, and he was able to, being that he was a great artist, he was able to draw what he saw under the microscope. And here on the right is a drawing here of the mammalian retina. And one of the things that he actually proposed was that when he looks at one animal to the next, a lot of these connections are very stereotyped. So for example, here you have the photoreceptors, so the rods and the cones that will connect within these plexiform layers to the bipolar cells. And then those bipolar cells will connect to these retinal ganglion cells down here. And then these retinal ganglion cells will convey information to downstream neurons. So what he observed was that these connections not only form contacts with with specific partners, but it seems to be that they form them in very discrete or organized fashion. And so this is actually very similar here in the retina, in the mammalian retina, to the invertebrate visual circuit. Here above, here you have your photoreceptors, and then those photoreceptors send their axons here into the lamina as well as into the medulla. And then there's interneurons that integrate that information and then send it to higher order processing centers. And it's very important for these neurons to be able to innervate specific layers, both in the lamina and in the medulla, in order to recognize their appropriate synaptic partners within those layers, and then to be able to convey that information to those downstream neurons. And being that this is today is the day after Valentine's Day, Ramon y Cajal was also a poet. And so when he looked at under the microscope, one of the little silhouettes that he has is that these synapses are actually what he described as protoplasmic kisses that appear to constitute the final ecstasy of an epic love story. And so hopefully throughout the talk, I'll be able to refer back to that in terms of how a neuron will eventually contact its fated synaptic partner during development. So I showed you here the mammalian retina as well as the invertebrate visual circuit. So there are different levels of circuitry throughout the animal kingdom. And so here we have C. elegans, which we have the connectome for C. elegans back in the mid 80s from Sidney Brenner. And so there are 302 neurons, 7,000 synapses, a very reductionist circuit, but it still has various functions that the worm has to do. On one of the other end of the spectrum is our own brain. Here we have billions of neurons and trillions of synapses, which is kind of mind-boggling to think about that there's organization within these circuits. But that's a little bit above my pay grade. I work here in the middle. So this is Drosophila. In the adult CNS, there's about 120, 150,000 neurons, tens of millions of synapses. Again, very large numbers, but as I'll show you, there's circuits within this larger circuit that we can actually interrogate to try to understand how they wire in with each other. So now let's talk about the stepwise process of how circuits form. So here we have 3 neurons. So very simple circuit with 3 neurons. This blue neuron has to form a contact with one of these, the red neuron or the green neuron. So there's initial process of axon pathfinding, axon guidance, different terms that have been used to describe this. But this axon will elongate, it'll navigate the environment, it'll respond and integrate multiple attractive and repulsive cues to eventually get to this neighborhood, this area. And so now it has to distinguish whether it's going to form a contact on this red neuron or this red neuron. And so through synaptic partner recognition, it's going to determine that it's going to contact the red neuron. And then once that contact happens, that ending elaborates and there's synaptic growth and it forms a fully developed axon terminal. And this is very simple, a very simple circuit. There are other neurons that will be contacting this neuron as well. And the blue and the purple neuron will also diverge and contact other neurons. And so it's a very complex circuitry, but it's very, very organized. And one common theme with all these processes is that there are cell surface proteins that need to integrate either these soluble signals or signals that are direct cell-cell contact in order to do each one of these processes. So I'll be mainly focused today on synaptic partner recognition. We do have some data that these genes that we're looking at are involved in these other processes, but I won't be discussing those today. And the second half of the talk, if I get to it, I'll be discussing these convergent circuits that there's multiple neurons that converge on a single target and how homeostatic or plasticity mechanisms allow for the regulation of activity there. So in the fly, there are 2 sort of big stages, developmental stages that have different circuits. So here is the embryo to the larval stage, and then during the pupil formation and pupil development, there's another circuit that forms that's completely different than the one in the larva. But the circuit that I'll be discussing, the neuromuscular circuit in the larva, actually forms here in the embryo. And so this is a video just to show you how dynamic the ends of these axons are. So these are all filopodia. And this is a live imaging. I forgot how many times we sped it up, but this is just to give you an example of how dynamic these filopodia are. And this is required because these filopodia and these axons need to survey as much of the environment as possible in order to eventually form contacts with its proper synaptic partner. And so here you can see these filopodia, they extend, they retract, they extend, they retract. And again, they need to do this in order to be able to survey as much of the environment as possible. So at the end of embryonic development, these filopodia will eventually form a contact and develop synapses at those contacts with specific partners. And so here, they'll form what we refer to as boutons. And within these boutons are these protoplasmic kisses, these synapses. There's 5 to 10 to 20 synapses within each bouton, depending on which NMJ you're looking at. And then the circuit is fully established at the end of embryogenesis. But then, as you can see here, the larva continues to grow and grow and grow. And so the muscles grow and the motor neuron ending also has to grow. So here's a fully developed NMJ from a particular motor neuron on a particular muscle. So this is at the end of third instar. Once it goes to pupation, this will all be broken down and you get a new circuit being formed there. So what does this actually look like in the larva? So here we have a third instar larva. So let me go back to here. This is the fully developed larva. So it has first, second, and third instar. This is a third instar larva. We dissect it along the dorsal midline. We take away all the guts, the fat bodies, and the various other tissues. And we're left with the body wall, which has the muscles, epidermis, there's sensory neurons there, as well as the CNS. And this structure here, the ventral nerve cord, analogous to a vertebrate spinal cord. This houses all the motor neurons. And let's zoom in a little bit here. And so these motor neurons here, you can see the cell bodies, will send projections out into the periphery and innervate specific muscles. And as you can see here, if we go back to here, you can see that these are repeated along they're mirrored along the midline so that you have mirrored circuits here, as well as anteriorly and posteriorly, so that each one of these can constitute its own circuit, which with the same neuron, with different neurons, but of the same type within each circuit. And so what this allows us to do, there's multiple circuits, it's stereotyped. We know the connectivity map of which neurons connect to which muscles. This allows us to go in there and interrogate if there are genetic mechanisms that allow for this hardwired map. You know, what are those genes that allow for this to happen? And that's what I'll be discussing today. And so here is a zoomed in of some of these NMJs. Again, these are the boutons that house the synapses. And I should mention here, we classify them into 2 different types. This is the type 1S in green, and the 1S neurons innervate multiple muscles. So these are divergent type neurons. And the 1B motor neurons innervate in a one-to-one fashion. So there are many more 1B motor neurons than there are type 1S. And so you have one-to-one connections and you have some neurons that connect to multiple targets. And I will be focusing on the 1S neurons that connect to multiple targets. And there are 2 1S motor neurons within each hemisegment. So each one of these hemisegments has 2 motor neurons that connect to groups of muscles. So let's talk about the problem that we're trying to solve here. So here we have one of these 1S motor neurons that connects to these dorsal muscles. And this particular 1S motor neuron connects to 5 muscles, but it has the potential to connect to 30 along, because there are muscles here on the ventral side that I haven't even shown that this neuron just bypasses. And so, but within this field of view, there are 8 potential targets, but it only forms contacts on 5. So we have, you know, this problem where there are 5 connections, but there are 3 muscles that it does not form a contact with. How does this happen? And again, this is very stereotyped when you look from one animal to the next to the next. So we can, you know, come up with different models here, different hypotheses. You know, one is maybe the muscles that the neuron does not connect with has a repulsive cue that says, hey, don't wire with me. I don't need your synapses. And so that can be a general mechanism to allow for this type of wiring. Another thing, which is not mutually exclusive, is that there are the same attractive cues at these respective muscles that are the targets that allow for that connectivity. So that it's the same cue here at this connection, same cue here at this connection, so on and so forth. And then finally, it's very possible that there are specific cues within each branch and within each muscle that allow for this connectivity. So here you have this B branch connecting to this B muscle. So there's one specific cue. There's another cue for this wiring pattern, another cue for this, and so on and so forth. And so whichever one of these mechanisms it is, it's very highly informative, since that will establish this type of connectivity. And so the question is, what are these molecules? And so that leads now to work that was done back when I was a postdoc in Kai Zin's lab. We collaborated with Christopher Garcia's lab at Stanford, and in particular, one of his postdocs, who's now is a faculty member here at UChicago, Engin Ozcan. So them being biochemists and structural biologists, we wanted to use our specialties to collaborate, because we're biologists and they're the biochemists, and we wanted to de-orphanize cell surface proteins. So as you probably are aware, most cell surface proteins are orphaned. We don't know what the ligand receptor combinations are. And so if we're trying to identify what these cues may be, we need to determine what the ligand receptor combinations are to try to interrogate those within our circuit. And so one of the striking findings from that study was the interaction between these immunoglobulin domain families. So these are the DPRs, there's 21 of these, and these are the DIPs, there's 11 of these. Some of these are very, very promiscuous, such as DIP beta, which has 7 binding partners. Others, such as delta, are very monogamous, so it only interacts with DPR12. And not shown here are some homophilic interactions. Some of these, such as DIP alpha, interact homophilically as well. And so what our model is that there's interactions across cells, such that the interaction, and we know that the interaction happens here at the first IG domain, that allows for this type of specificity and connectivity to occur, such that the expression of these cell surface proteins will allow for the identification of that particular cell type. And there's been various studies now, even from Barry Honig and Larry Shapiro, showing what the affinities of these interactions are, as well as even some of the structures of these, to try to determine what the mechanism is to allow for this type of promiscuous interactions. So if we think that this is a model, we can predict several things. So first is that these DPRs and DIPs will be expressed in specific subsets of neurons. If they're expressed ubiquitously, then how do you get particular specificity in the connections? Secondly, is that the cognate DPR DIP pairs will be expressed in the synaptic pairs. And third, that if you lose some of these cell surface proteins, you likely would get connectivity defects. I put a little asterisk here because if you look at this interactome, there's a lot of overlap. You could potentially get multiple or co-expressed DIPs, for example. And so if you get that, there may be some redundancy or some combinatorial interactions that allow for that specificity to happen. So if you get rid of one DPR or one DIP, you may not reveal the phenotype. And so that becomes a little bit more complicated, but since we have the interactome map, we can try to interrogate those types of questions. And then finally, we can try to skew connectivity by ectopically expressing some of these DPRs and DIPs. And I'll go over all 4 of these during this first half of the talk. So first, we wanted to look at the expression of these DPRs and DIPs within the motor circuit. And so to do that, we collaborated with Hugo Bellin's lab at Baylor. And from there, they have a sort of an army of people over there. And so we have now DPR DIP GAL4s that allow us to monitor the expression of all of these genes within the motor circuit. And this was work that was mainly done by Yupu, as well as some undergrads that helped them. And so since we have the connectivity map, we're able to drive GFP or any fluorophore under that GAL4, which will then give us where the DPR or DIP is expressed. Here's the musculature. So for example, if a DIP or DPR is expressed in a particular motor neuron, we know the map. So then we just have to look at the NMJ and say, oh, the NMJ lights up with the fluorophore. So then motor neuron 4, so this is the motor neuron that connects to this, which is the 1B subtype. That one expresses a particular DPR and DIP. If we have this type of expression, we'll see the NMJs on all these muscles, and this will correspond to one of the 1S motor neurons, which we call the ventral common exciter, because it innervates the ventral muscles. So that's what Yupu and his peers did, was to go through the full DPR and DIPs, all DPRs and DIPs, and just to catalog their expression. And this is the culmination of a lot of their work. So there's a lot of data here, but general things that we can extract from this are that here at the top, I should mention, on the left are the genes themselves, and on the top are the motor neuron types. So here you have your 1B types, and then here you have your 2 1S motor neurons. And so just glancing at this, the dips here seem to be much more sparsely expressed in motor neurons. Some of these, like alpha, are expressed in 2 motor neurons. Others, such as delta, are expressed in 1, and even here, eta is expressed in 1 of the 1S motor neurons. So there's very high specificity, but others are sort of sprinkled here and there. And I should mention the different colors. What they also were able to find is that there's some differences in the anterior-posterior expression of some of these DPRs and dips, as well as some temporal expression. Not too many temporally, but there is some temporal expression. DPRs this is one thing that we're still trying to wrap our head around, but DPRs seem to be much more widely expressed in motor neurons. So as you can see here, some of these, like DPR1, are expressed in every motor neuron. And so I have some thoughts about that we can discuss at the end if you'd like. But importantly, each motor neuron, so if we look down each column, each motor neuron expresses a specific subset of DPRs and dips. So no column will be alike. So that could potentially be some of these identification tags or serve as a code for that particular motor neuron. And so that's the premise of the model that we're interrogating. So today I will focus mainly on DIP alpha and its interactors, being that they're expressed only in the 1S motor neurons. And we also had multiple tools, genetic tools, to allow us to analyze this. So let's see what that looks like in terms of the expression. So again, they dissect third instar larvae, and we used a few different reporter lines, but here I'll just show you a sort of a UAS GFP reporter. So you can look within the ventral nerve cord, and then you can see a bunch of neurons here. Remember, I told you that alpha expresses in 2 of the motor neurons, and these other cells here are actually interneurons within the ventral nerve cord. And being that I've looked at this for so many years, these here in the midline, these are one of the motor neurons here. This is one of the 1S motor neurons. And so again, they look out in the periphery, and then now we can start to figure out what those motor neurons are. So this is these are the dorsal muscles. You can see here in red are the 1B motor neurons, and in green is wherever DIP alpha is expressed. Here we have the alpha driving GFP. If we get rid of the red, here we can now see this is the stereotypical 1S innervation pattern for the ventral common exciter on these ventral muscles. So we know DIP alpha is expressed in that neuron. If we look at the dorsal muscles, now again, we see that here in the 1B motor neurons, DIP alpha is not expressed, but here now you see expression, and you can see here that there's one axon within this nerve, and this one axon is sending out branches to its respective partners. And so if we get rid of the red, again, this is the very typical innervation pattern of the dorsal common exciter. So we have DIP alpha that's expressed in these 2 motor neurons. But this is at the 3rd instar, long after the circuit has been established. So we went back earlier in development to see when DIP alpha starts to be expressed, and not just expressed, but to see where it localizes to the terminal. So here we have stained, these are all the motor neuron filopodia, not just for the 1S motor neurons, but all motor neuron filopodia. And you can see here, and these are some of the growth cones. You see that there's multiple filopodia coming off of those growth cones. And so here now we have a fused DIP alpha to GFP, so we can look at the localization of DIP alpha. And this is an endogenous tag. And so what we can see here is that DIP alpha localizes to a subset of these filopodia, which is what we would expect since it's not expressed in all motor neurons. And so if we get rid of the red, here now we can start to see this is the axon trunk of the 1S motor neuron, and these are some of the filopodia coming off of that axon. So DIP alpha is expressed there at the stage where it should be if it's going to be involved in wiring, and it localizes to the particular structures that actually will be contacting those muscles. And I should mention here, so I have a schematic here. This is the growth cone. These are the filopodia from that growth cone, and the muscle has similar filopodia called myopodia. So now with this, we can say that DIP alpha is localized here to the filopodia. So now we want to look at the postsynaptic side. So being that we have the interactome, we know DIP alpha interacts with 6 and 10, and this is in vitro. And so the question becomes, are either one of these expressed in the cognate synaptic partner, which would be the muscles? And I'll tell you that DPR6 is not expressed, and so I'll show you some data for DPR10. So this is DPR10 GAL4 driving GST. This is at stage 17, right after the circuit has been established. And as you can see here, it's expressed in a large subset of muscles. Some of these are not, they're not labeled here, but there's muscles underneath here that don't express DPR10. So for DPR10, we did not have an endogenous tagged DPR10, but we do have is a transgenic line with DPR10 tagged. So we want to see where DPR10 is able to localize. So you can't see this here, but let me go to the next slide. So if we express DPR10, and this is at an earlier stage, right before the circuit is established, you actually have DPR10 that's expressed in a smaller subset of muscles at this earlier stage. But importantly, here you can see the myopodia that I was discussing here in the schematic. And you see DPR10 there within that myopodia, here more clearly visualized here. So DPR10 localizes to the tip of that myopodia, and I can add that here to this schematic right here. So now, as a quick summary for this part, we have DIP alpha that's presynaptic, we have DPR10 that's postsynaptic, and then the question just becomes, what are they doing there? And so we knocked out each one of them, and then we looked at the connections of the motor neurons onto respective muscle partners. And so this is at the first instar, right after the larva hatches, right after the circuit is formed. So here we have the 1B motor neurons in red, and then we have in green, we have the 1S motor neurons. If we remove DIP alpha, I should mention, we're looking at muscle 4 here. This is one of the muscles, one of the 30 muscles within the body wall. So if we look at DIP alpha mutants, we see that muscle 4 is not innervated by this 1S neuron. It actually just bypasses it, and it goes on to innervate other muscles, but it does not innervate this particular muscle. And so that is very clearly shown there. And importantly, DPR10, if we remove DPR10, that also leads to the exact same phenotype, again, suggesting that there's trans interactions between these two cell types that allow for this to happen. We've now gone on to show we have mutants now that actually just break the interaction, and we can actually show now that we cannot rescue the phenotype with those mutants as opposed to a wild type. I'm suggesting that it's the precise interactions between DIP alpha and DPR10. So we can quantify this. Here we have, again, the schematic, alpha pre, 10 post, and this is muscle 4 innervation. Muscle 4 is innervated by the 1S motor neuron about 80% of the time. Other muscles are innervated 100% of the time by the 1S motor neuron, but this particular muscle is 80% of the time. If we remove DIP alpha, that leads to no innervation. So this was, we got very lucky here. I mentioned that these are potentially redundant or combinatorial interactions may be required, but for this particular connection, we got very lucky because removal of DIP alpha led to absolutely no connections forming on muscle 4. And when we removed DPR10, that led to the exact same phenotype. So no connections formed on muscle 4. And we have a bunch of tools that we can then use to show that DIP alpha is needed pre and DPR10 is needed post, and that's what we did here. So in a DIP alpha mutant background, we can reintroduce a wild type DIP alpha, and that leads to complete rescue of the phenotype. And for DPR10, what we did is an RNAi knockdown approach. And if we express that in the muscles, that also leads to the phenotype. So now we have here what we think is a bona fide trans interaction that allows for that connectivity of that motor neuron onto that specific muscle. I didn't show you the data, but there's other muscles that lack partial innervation by that same neuron. And then there's other muscles that absolutely look wild type. And I'll get back to that later in the talk. And so here, if we remove DIP alpha or DPR10, again, we remove this muscle 4 connection. So now we can ask, so these are necessary for connectivity, but are they sufficient to drive connectivity? So that's what we did next. And so here we have a 1S motor neuron that will innervate both of these muscles, and this is muscle 7 and 6. And so this 1S motor neuron expresses DIP alpha. And so what I'm going to try to do now is to overexpress DPR10, the partner for DIP alpha, selectively on this muscle, and to determine whether we see normal connectivity or if we can skew connectivity such that now that 1S neuron only connects onto muscle 6. So it becomes very sticky on muscle 6 and it's not able to innervate muscle 7. So this is what we did in this next round of experiments. So here we have muscle 7 and 6. This is a little more difficult since we don't have the 1S labeled, but in bright magenta or bright red here is the 1B motor neuron, and in sort of this more clearly seen here, in this lighter magenta is the 1S motor neuron. And you can see that the 1S is on both muscle 7 and muscle 6 as well. So we have a driver that will only express in DPR muscle 6. And so what we did there is express DPR10. And what we can see is now the 1S motor neuron completely now shifts its innervation only onto muscle 6. The 1B motor neuron still connects to muscle 7 and muscle 6, but now the 1B shifts its connectivity to muscle 6. So possibly becoming too sticky onto muscle 6 and then forming all its synapses onto there. So this was pretty exciting. Now we're trying to do it with various combinations. For example, if we know some of the dips and the DPRs that are expressed here within the 1B motor neuron, to see if we can do something similar by overexpressing some of their interactors within this muscle. So now let's take a step back. I mentioned that the dip alpha phenotype is not all or none. There are some muscles that still have innervation. So going back and looking at our expression map, dip alpha is expressed in these two 1S motor neurons, and we have various other dips that are also expressed in those 1S motor neurons. So gamma, epsilon is expressed only in one of them, zeta is expressed in both, eta is in one, theta, so on and so forth. So what we are now trying to do is, we think that these are acting combinatorially such that you need multiple of these interactions to get a full connectivity map. And so we're simultaneously knocking out these DPRs and dips. And here I'm going to show you some preliminary data for, this is the innervation of muscle 13 by one of the 1S motor neurons, which is that ventral common exciter. So this is one of those muscles that is absolutely 100% of the time innervated by the 1S motor neuron, as you can see here. In a dip zeta mutant, so again, dip zeta is expressed in both of these, you see nothing. So it's still innervated. In a triple mutant, so this is actually, eta is not expressed here, but this is a line that we got from Larry Sapersky. It's a dual knockout for eta, theta, since they're in such close proximity to each other. But here we can say that this is a double mutant for theta and zeta, again, fully connected. When we look at a dip alpha phenotype, now you start to see a slight decrease in the innervation of muscle 13. So you lose about 30 to 40% innervation. Now we can combine this alpha mutant with this triple mutant to see what happens. And so there you get even a larger decrease in connectivity. And so what we think is happening again is that there are combinations of these cell surface proteins expressed in particular neurons that then will be binding to the cognate DPRs on the synaptic partners to get the full innervation that we see for some of these. And so that's where we're at here. We're making quintuple, sextuple, septuple mutants. Thankfully, they haven't been lethal yet, possibly because removal of a small subset of connections doesn't completely screw up the animal, which is, for us, that's exciting because we can actually go in there and look at specific connections, both because we have the expression map and the interaction map. A quick summary for here. So here we show that alpha is here presynaptically and DPR10 is postsynaptically. And the interaction between DPR10 and dip alpha seems to be a specificity cue to allow for motor neurons to recognize a subset of their muscle partners. We can skew connectivity by overexpressing some of these DPRs and dips. And importantly, and this is for us, for me at least, one of the more exciting things is that we can now start to do loss of function of multiple DPRs and dips to try to interrogate the system. And I think this is part of the reason why a lot of these cell surface protein identification cues haven't been found in a tons of screens that for example the Goodman labs and so on and so forth have done throughout the years. Because if you remove one identification tag, there's a good chance you're not going to see a phenotype until you start removing multiple. And in those types of screens, it's difficult to target multiple genes simultaneously. And so now we can systematically start to look at this. So now for the next 10 minutes, I'm going to go over the functional side of one of the projects that we're doing. And so I'm going to switch gears. It actually, I'm not going to show it to you. I'm not going to sell it to you how we found this out, but it actually stems from part of this losing connectivity on specific muscles. And so this I'll be discussing is synaptic plasticity in convergent circuits. So, and this is work by Yifu Wang. And so, you know, a common circuit motif in almost all circuits is that there is convergence of neurons onto one target. And so the activity of these neurons will, you know, if that leads to enough activity within this postsynaptic neuron, you can get reach super threshold, and then this neuron can fire. And so there'll be stimulus that allows for these neurons to be activated, and then it'll relay it downstream. And so what we asked here was, you know, if one of these neurons, one of these pairs is ablated or undergoes degeneration, what happens to this circuit? Are there compensatory mechanisms that allow for this circuit to maintain the normal level of activity as if this neuron was still there? And again, this stems because we actually looked at some of these mutants, and we did not see that, we saw that there was some compensation. And so, you know, in the musculature, in the neuromusculature here, we have multiple neurons that will be innervating the same muscle partner. And so we can more easily interrogate that here. You know, there's a lot of labs that have gone in the neuromuscular junction and looked at plasticity, but they mainly look at plasticity between the synaptic pairs, not partners, not between the innervating partners. For example, if you knock down glutamate receptors postsynaptically, you're able to see a homeostatic compensatory upregulation of neurotransmitter release from the presynaptic neurons. Again, labs from Gray Davis and Dion Dickman and so on and so forth. And so here we're going to interrogate multiple muscles. So each muscle is innervated by a particular 1B motor neuron. I mentioned that these are one-to-one. And remember that the 1S neurons innervate multiple muscles. But each muscle receives a unique pair of 1B, 1S motor neurons. So we're going to be interrogating muscle 6, 12, and muscle 4. So again, each one of these is a separate 1B motor neuron. The muscle 6 and muscle 12 share the same 1S neuron, but again, the pair is unique. And so what we can do here is ablate these 1S motor neurons, and then just look at the circuit, look at both the structural morphology of the NMJ from the 1B motor neuron, as well as the function. So we can stick electrodes in this muscle to determine if there's any compensatory changes that happen. And so here I'll show you that we can easily ablate these motor neurons. And so this is a driver that we generously got from Richard Mann's lab. And so this is actually derived from the DIP alpha locus. And so this A8 specifically expresses within 1S motor neuron. And so here you can see the cell bodies within the ventral nerve cord, and then you can see the NMJ arbors in the periphery. And so we can drive these, we can drive Reaper and HID within these 1S neurons, and then we can see that we completely lose the cell bodies as well as the NMJs. And so this allows us to very easily ablate the 1S motor neurons. And so then the question becomes, what happens to the neighbor? What happens to the 1B that's still left on that muscle? So here we have a control. This is muscle 4, where you have the 1B and the 1S here. We ablate the 1S neuron, and what we see is that the arbor actually expands. So we have more boutons. So here we can just go in there and just count individual boutons. And so here you can see in the darker colors are the controls for each one of these respective muscles, and sort of these lighter colors are the 1S ablated experimental samples. And so here we see on muscle 6, you see an increase. On muscle 12, you see an increase. And on muscle 4, you see an increase in the NMJ size. And so this is suggesting already that there is something happening on the 1B motor neuron that allows it to detect the loss of that partner. You know, whether it's through the muscle, whether there's other signaling mechanisms, we can discuss that later. So now we wanted to look to see, you know, what happens functionally. So we see a morphological change, but that does not necessarily mean that there's a functional change there. And so what we did is to measure the spontaneous and evoked neurotransmission. And here we're looking at muscle 4. And so we know that one of the things that happens is there's basal release of neurotransmitters from the presynaptic side. You know, it's been shown that spontaneous neurotransmission is very important in synaptic development. So we can stick an electrode here into this muscle and then record the miniature excitatory potentials that are a result of one vesicle being released of neurotransmitter. What we can also do is evoke an action potential here presynaptically and measure the postsynaptic side, the excitatory response, based on that bolus release of neurotransmitters there. And so that's what we did. We measured the spontaneous and the evoked transmission. So the Isikoff lab back in 2007 showed that the 1B motor neurons are released much more, the frequency of release is much more compared to the 1S neurons. And so it's a 2-to-1 ratio between the 1B and the 1S neurons. And so if we ablate the 1S neuron, we would imagine that we would see about a 33% decrease in the spontaneous release if there's no changes here to the 1B. And so we went in there and we checked this, but what we see is that there's absolutely no change in the spontaneous release of these 1B motor neurons when you ablate the 1S. So again, here, this in muscle 6, in muscle 12, you actually see a slight increase, and in muscle 4, again, there's no difference. And so what this tells us is that that 1B motor neuron is elevating its spontaneous release here. And so now there's a functional compensation, at least at the level of spontaneous release that's happening in these 1B motor neurons. And so now let's get at the evoked neurotransmission, which is actually needed to contract those muscles. And so to do that, we needed to tease apart the 1B response from the total response. And so to do that, we used GCAM combined with electrophysiology to determine when the 1B was firing as opposed to when the 1S was firing. Each neuron has a different super threshold, so we can sort of tinker with the stimulus to activate the 1B or the total 1B plus 1S. So here we have the 1B and the 1S that are firing. You can actually see some spontaneous release every now and then. But here you can clearly see that the 1B and the 1S are firing. So now we're gonna lower the stimulus threshold, and that's only gonna fire the 1B. So if we have an electrode here placed in this muscle, we know that the excitatory potential is a result only of this 1B because the 1S is not firing. So here are the 1B plus 1S at the top, and then the 1B alone here in a control. This is not ablated, this is a control. So what we can do is take this ratio, the 1B over the 1B plus 1S, and then determine what the percentage of the total response from the 1B. So here on muscle 6, we have about 50% of the 1B that the 1B is contributing to the total. On muscle 12, it's about 30%, and on muscle 4, about 60%. So the one thing that we already noticed here is that the 1B response is different on each one of these muscles. And again, this is in a control. So now what we can do is let's ablate the 1S motor neuron, see what happens to the evoked activity. So here on muscle 6, we see that the 1B, this is the total response, 1B plus 1S. As we can see here, there's definitely a decrease here on muscle 6, 12, and slightly on muscle 4. But what we can do is compare it to that ratio. Now, I mentioned this ratio here. We can compare it to that ratio because that's the 1B response isolated. And so when we do that, we see that on muscle 6, there's a slight increase compared to that ratio of the 1B motor neuron. On muscle 12, there isn't. But on muscle 4, there's a significant upregulation of evoked activity on muscle 4. So suggesting that there's a target-specific manner of how the evoked activity is being compensated for. Both of these muscles show the structural and the spontaneous, but not the evoked. So this is just a summary of what I just showed you. You ablate the 1S, you see upregulation in the 1B. And at the same time, as we were doing this, Troy Littleton's lab actually showed on muscle 1 that the same thing happens there, where you get an evoked response, an evoked compensation when you ablate the 1S motor neuron, which is nice when somebody else independently confirms your data. And so in another minute or two, I'll just quickly go into some, what are some of the mechanisms that allow for this to happen. I'll kind of skip this. We inhibited the evoked response of the 1S neuron, and we see that that is able to allow for some compensation to happen. So the evoked response has something to do with the compensation in the 1B. But one other thing that was pretty interesting here is how the 1B neuron knows how much to compensate. So I showed you this similar schematic early on, where synaptogenesis happens here in the embryo. And so what we wanted to determine is when does the 1S neuron die when we actually ablate it genetically? So we went earlier in development. And in the first instar, we noticed that the cell bodies are still present. The NMJs are undergoing degeneration, but what this tells us is that the neuromuscular circuit is actually forming, and then you get degeneration. And so in our model, it's not that we're killing off the neuron before it's able to innervate its target. And so ablation happens after synapse formation. And so what we're suggesting here is that early in development, you get both the 1B and the 1S that form synapses onto this muscle, such that this creates a homeostatic set point that then perturbation can reference down the line. So if you ablate this 1S neuron, this homeostatic set point is going to signal back to the 1B motor neuron so that it knows how much to compensate. And if you have this 1S neuron ablated, so this is going to be a set point. If it's ablated before synaptogenesis, this set point is going to be only the 1B response and not the 1B plus 1S. So let's cut to the chase. Because alpha, the phenotype for alpha does not allow the motor neuron to form a synapse onto muscle 4, we can interrogate that with that genetic background. And what we see here now is that the NMJ morphology is not expanded, so we don't see an expanded arbor on muscle 4, unlike the ablated 1S motor neuron after synaptogenesis. The frequency of spontaneous release also decreases, which is what you would expect if there's no compensation. And then the evoked activity, there's a slight increase, but nothing to the extent of when we just ablate the motor neuron. So what this is suggesting is that there, again, the 1B plus 1S contribute to this homeostatic set point, such that when we ablate the 1S motor neuron, whether it's the evoked activity or other mechanisms that are required to allow for the 1B to respond, that's one thing that we're trying to figure out now. But that 1B is absolutely, some of the 1B motor neurons are absolutely able to compensate both the morphology and the function. And so what we're trying to do now is to determine, again, what are these signaling mechanisms that allow for this plasticity? What happens to the machinery here at the 1B NMJ to accommodate for these functional changes? And, importantly, is there a critical period for this, or is the 1B NMJ able to respond at any time during larval development for those perturbations? So with that, just thanks to everybody in the lab, some collaborators. Larry shared a few of their DPR and DIP mutants, Hugo with his GAL4s that allow us to do the expression screen. And again, we're continuing our collaboration to look at DPR DIP interactions, as well as interactions between DPR DIPs and other cell surface proteins, as well as for funding. And with that, I will take any questions. I cannot believe I got through that. Holy Jesus. Okay.
Impressive. Okay, I wanted to ask a quick question. I had noticed in one of your DIP alpha mutant slide, only 80% of the controls had connectivity to, I think, the muscle 4. Is that just like the normal, like you usually normally see some just don't have connectivity, and does that like affect the EPSPs? Yeah, can you just say a little bit about that?
Yes, so one of the reasons that I think we got lucky with alpha on muscle 4 is that is one of the muscles that is not always innervated. And so that suggests to me that, throughout evolution, the 100% innervation of muscle 4 is not required for function, for normal crawling, feeding, so on and so forth, foraging. And so maybe all the, before maybe it was innervated 100% of the time, but now it lost some of the cell surface proteins. And so it just doesn't fully form that connection onto there. But yes, in other animals, it's the average is about 80%. So you will always see that. And absolutely, the function, the function of that muscle, if you look at it in a control that doesn't have the 1S, that looks like the DIP alpha phenotype, because that it just never forms it. It's not that it forms it and then it retracts it. It just never forms it. So the function is perturbed at that muscle.
Gotcha. Thanks.
Yep.
Okay, Richard Mann has a question. Does the homeostatic compensation of 1B when 1S is ablated require DIPs or DPRs? That's where we're going. I don't know the answer to that question yet.
I will do a little teaser here. Is during when two motor neurons come onto one muscle. So a lot of the images that I showed you are at third instar when they're separated. The muscle is huge, and so the boutons are very separated from each other. But if you look earlier in development, when the circuit's being established, these boutons are, and not just even the boutons, even before the filopodia are almost overlapping and on top of each other. So there's potential for 1B and 1S endings to be able to do cell-cell contact. So I'll leave it at that and just say that we're trying to examine whether there is some interactions between the 1B and the 1S motor neurons that allow for this to happen.
Cool, thanks. Also, I see Oliver Hobart has his hand raised, but I don't know if we can prompt video. So if you could put your question in the Q&As, then I can ask it. Okay, from an anonymous attendee, does the animal behave normally after you delete DPRs and dips?
So we've done we have the great Ellie Heckscher next to us, who does a lot of behavior. So we've done some just very gross larval locomotion assays, and you definitely see them see some defects with specific dips. We've done the dips, and I think that just depends on how much loss of connections you have with each respective dip. What I want to do now is to do some of these triple, quadruple, combinatorial loss mutants within those assays to see what happens to the locomotion.
Cool. There was one, I think, in the chat to the panelists. How exactly were the neurons ablated? That'll probably be pretty quick. But
yeah, we have these cell death genes, Reaper and HID, and they've been used throughout for a long time to induce ablation. So you just express it in your specific cell type.
I also had another question about during the targeting phase, are the ventral muscles usually innervated before the dorsal ones? And because they're presumably like the axon terminals are growing from the VNC, right? So do the ones that will eventually innervate the dorsal muscles, do those try to innervate the ventral and then realize, oh, that's not my target, and then they just like fly past it? Or did they never even try to synapse onto the ventral neuromuscular junction?
So in relation, let's go back to the first one, ventral versus dorsal. So we're trying to do that now. There's some old papers showing that the ventral versus dorsal, there's no precise. It seems that the dorsal muscles are actually the ones that, within the dorsal group, the dorsal muscles, the most dorsal are the ones that innervated first. There's pioneer neurons that actually go there first to then allow the other motor neurons to follow. So it potentially may be at the level of dorsal to ventral, but I don't think I think we still need to explore that a little bit more with more current techniques. And so yes, to the other part, the dorsal motor neurons have to bypass the ventral muscles in order to get to their dorsal field. And there are filopodia that have the potential to interact with muscles on the ventral muscle field. That's one thing that I'm actually trying to figure out how to do, because I would love to be able to see what cells along the way those motor neurons are contacting. Sort of, obviously doesn't form stable contacts, but in passing, what cells does it form contacts with? But yeah, there is the potential to form connections to there, and you can get ectopic innervation of some of those muscles sometimes in different backgrounds.
Cool, thanks. Okay, this question comes from Patricia Cooney. Do you think the homeostatic mechanism of 1B increasing their firing when 1S is ablated is dependent on local muscle to motor neuron interactions at the NMJ, or involve a closed loop circuitry like muscle output to proprioceptors to PMNs?
Yeah, that's what we're trying to get at now. The simplest would be that, in a lot of the other assays that they've done for plasticity, again, if you inhibit the postsynaptic glutamate receptor, you see an upregulation of release presynaptic. So, you can imagine that, the removing the 1S neuron, that muscle senses that there's something wrong there. And so it could very simply be that the muscle senses that maybe it's similar in mechanisms to the what I refer to as presynaptic homeostatic plasticity, where the muscle then tells the presynaptic motor neuron, hey, something's wrong. You need to just spit out more neurotransmitter. It could be as easy as that. Our data is suggesting that it's not. There's other cell types that I think are functioning there, not proprioceptive, but we think that it's either, it may contribute, the muscle may contribute, but I think it is either axon-axon or potentially axon-glia interactions that are happening there.
Cool.
Great question.
I don't see any more questions, and I also don't see the.
I can talk to Oliver later. Well, I talked to him this morning, but we can chat over email.
Maybe he like accidentally raised his hand.
Yeah.
Sarah, is there the link to the mingling session for the chat?
Yes, 1 second. I'm not used to people ending early, so give me 1 second to pull that up and put it in the chat.
I'm sorry, I went so fast. Gosh. Oh.
Oh yeah, Oliver said it was an accident.
Okay. I guess I was so clear that he didn't. So I also have to click on another link, right?
Yes, I believe so.
Okay.
Yes, if everyone can please just hold tight, I'll post the link in the chat. Thank you.
How's the weather over in New York, Abby?
Oh gosh, it's cold.
What is cold?
I think it was like 23 when I'd walked
Okay.
today. So
Yeah.
I think it'll warm up by the end of the week though. So
Yeah.
it's good.
It's supposed to begin a big storm though. So I'm assuming that once it comes through here, it's going to go towards you.
Yeah. Okay. There's
Okay.
the link for the mingling session. So if you'd like to attend, I guess we'll head there now.
Sounds good. I'll see that.
Thanks, Robert, again.
Hmm?
I said thank you.
Oh yeah, no problem. Thank you. Appreciate it. Okay, I have to end this.